out of what I tell you in the next 2 days, but if you do only learn one thing, this is it. And this is something that you need to know for the rest of your career if you're going to be working in a laboratory. And it applies across the board, no matter what you're working with, whether it's infectious agents, hazardous chemicals, radiological material, it doesn't matter. It's extremely important that you understand what a risk assessment is, who should be doing a risk assessment, and how to do a risk assessment. So obviously, the word risk implies that harm, injury, or disease is going to occur, or could occur. So when we talk about working with biohazards, we're talking about specific risks, such as infection, might be associated with working with these materials. So we do what we call a risk assessment, and this is performed to determine the appropriate containment, which includes the facilities, the equipment, and the work practices that should be used to reduce or prevent exposures, which therefore reduce the risk. The principal investigator, or the supervisor in the lab, they have the primary responsibility of performing the risk assessment. However, and this is the most important statement, every individual who is doing the work needs to be involved in this process. I don't know, how many of you have actually worked in a lab, in a research lab? Okay, good. How many of your principal investigators did bench work? Okay, that's pretty common, very, very few. Most of them write grants, go to meetings, do the important stuff. They get the money to continue doing the research. It's people like us that do the bench work. You guys know what the actual risks are because you're the ones that are actually doing the work. The PI doesn't always know that. The PI thinks they know because they think they know what you're doing in the lab, but they don't always know what you're doing in the lab. You need to tell them what you're doing in the lab. They write a procedure and they think this is exactly how it's done, and they should give that to you to review, and you should say, yes, this is what we do. However, in this step, we also do this, and we need to make sure we're wearing X, Y, and Z PPE because there's a potential for exposure in this step. They might not know that because they don't do it every day. They might have 20 years ago when they were just like you, but they don't do it anymore. Things might have changed. New procedures might have come up. The SOP might be different now. The only one who knows what you're doing every single day is you. So you have to be involved in this process. Not to mention, you're the ones at risk, not them. They're sitting at a desk in their office. So you have to make sure that the risk assessment is actual and factual because you're the ones that are putting yourself at risk when you're working with hazardous materials. It's the same thing when you're working with chemicals. It's the same thing when you're working with radiological materials. You need to make sure that when you do a risk assessment that what you get out of it is valid and actually mitigates the risk, that you have the right engineering controls, that you have the right work practices in place, that you have the right PPE available. Because if you don't, this process is worthless. If someone who's sitting in an office in another building does your risk assessment for you, you might as well not even do it. So you need to make sure that you are always a part of a risk assessment that you're going to be working on a project for. And when you're doing a risk assessment, especially for biological agents, these are the things that you need to take into consideration. So when you're working with an agent, you want to look at what's its pathogenicity. So what's the ability of the agent to cause disease? The more communicable it is, the more severe disease it causes. Obviously, causes more disease, higher risk, right? You're working with Ebola virus, higher risk. You're working with E. coli K12, no risk at all. You're going to have much different risk assessment there, right? What's the route of transmission? Is it a parenteral risk? Do you have to inject it into yourself? Is it a needle stick risk? Or can you get it via aerosol route? Those are very different. So aerosol, much higher risk. If you actually have to ingest it, there's a lower risk. Okay, so it's a little bit different risk factor. How stable is the agent in the environment? If you have a spill and the agent can live for weeks in the environment, the risk is a little higher. So things like Bacillus anthracis, that can live for a long time. Hepatitis B. Hepatitis B virus can live for 14 days outside of the body. It's very stable in the environment. HIV, that can only live for about 24 to 48 hours, so it's a little bit lower. That all affects risk factors. The infectious dose of the agent. If you only need a few organisms to get sick, the risk goes way up. If you need millions of organisms to get sick, then the risk is a lot lower. How much agent are you actually working with? I mean, if you're working with a clinical sample, a lot of times the risk is a little bit lower because the infectious dose or the concentration of the agent present is lower. If you're talking about, a needle, something that might be on a needle, you don't obviously have a lot of agent on the surface area of a needle. If you're talking about a liter of blood, you might have more agent present. If you are concentrating virus, you've got a lot of agent present, so you're dealing with a lot more agent there. What's the origin of the infectious material? Is it something that we work with all the time? Is it something that is native to the U.S.? Is it, something from Africa that we don't see very often, that we might not have a vaccine for? And that goes into the next one. What's the ability of effective vaccinations and antibiotics? Do we have antibiotic treatment or antiviral treatment available? Or can we be vaccinated for it prior to using it? And then what's the experience and skill level of the people that are going to be working with it? And that also should include people who are just going to be in the general area. If you're working with animals, you want to take into account your animal care staff. If you've got routine people who work in the laboratory area, like housekeeping people and maintenance people, make sure they're trained and they understand what's going on in the lab and what the risks are to them as well. There is a reference which, if you're working in a biomedical lab, you should all be very familiar with this reference, and this is the Biosafety and Microbiology and Biomedical Laboratories, otherwise known as the BMBL. This is put out by CDC and NIH. If you just Google BMBL 5th edition, it will come up. I also have the link at the end of the presentation. This is kind of the Bible of biosafety. There's a whole chapter on risk assessments in there. Very good information. But there's also tons of information in this book. It's available free of charge online. You can also print it if you want it at no charge. But you should become very familiar with this book. This is what it looks like. So you will hear, the more you start working in a lab, things called risk groups. And many years ago, CDC and NIH got together and decided we need a way to classify infectious agents to help us decide how risky they are to work with them. So they basically went through every infectious agent that they knew of at the time, and they categorized them into 4 risk groups. This is what they called them. And they're based on their relative hazard. So they took all those things into consideration that we just talked about and put them into these 4 risk groups. So risk group 1 is the lowest, risk group 4 is the highest hazard. Risk group 1 are agents that are not associated with disease in healthy human adults. Okay, so this is the lowest risk group. When you take like a high school science class and you do a little bit of microbiology and you streak some plates, you're usually using a risk group 1 agent, something that's not going to make you sick. E. coli K12, your yeasts, things that the yeast that you make beer with, risk group 1. Most of the E. colis that you do recombinant DNA work with are in risk group 1. If we move up a level to risk group 2, these are agents that are associated with human disease, but rarely serious, and preventative or therapeutic interventions are often available. So we've got antibiotics or antiviral medication. Many times we've got a vaccine available. These are things like Staph aureus, Salmonella, and all human source material. So human blood, human cell lines, even if you have an established human cell line that you purchase from like ATCC, these all are risk group 2 organisms. Okay. The reason that we put all human source material in risk group 2 is because of the potential for bloodborne pathogens. And when you take your bloodborne pathogen training, you'll learn about the bloodborne pathogen risk. Most of these agents, they're going to make you sick, but in a couple of days, you're going to feel better. You get salmonella, you're going to take some antibiotics, you're going to be pretty miserable for a few days in the bathroom, vomiting, diarrhea, but maybe dehydrated, have to go to the hospital, but you're going to recover. No lasting effects, except maybe mentally, but you'll be okay after that. Risk group 3, though, these are agents that are associated with serious or lethal human disease. There may be preventative or therapeutic interventions available, but these are usually transmitted via aerosol route. Okay, risk group 2 agents were usually transmitted by needle stick or by contact wound. So we usually have to have some sort of direct contact or ingestion or needle stick route. Okay, mucous membrane, something of that nature. We get up to risk group 3, now we can transmit via aerosol route. So a lot easier, either person-to-person transmission, or if you're working in a laboratory setting, you can transmit it to yourself occupationally via aerosol. So now we're talking about things like Mycobacterium tuberculosis, Bacillus anthracis. There are also some risk group 3 agents that aren't aerosol transmission, things like West Nile virus, St. Louis encephalitis. So they're just a little more serious disease type agents. Risk group 4 agents, these you do not want to, you want nothing to do with these. These are things that are likely to cause serious or lethal disease, but unfortunately we don't have treatments or vaccinations available for these. These are things like Ebola virus, your hemorrhagic fevers. I would recommend staying away from them because you probably won't survive very long. Okay, so we've talked about biohazards and we've talked about biosafety.